Hello everyone. This video will cover the manufacturing process for mesenchymal stem cells, MSCs, and also highlight some of the key challenges we are facing. Mesenchymal stem cell therapy has high potential for tissue regeneration, combating incurable diseases, and anti-aging. However, one of the main bottlenecks in mesenchymal stem cell therapy lies in manufacturing consistent batches that produce our desired clinical outcome when delivered into the body. Despite numerous number of clinical studies since 2004, only 15 MSC-based products have been commercially approved today. A manufacturer of MSC has to take into account the source of MSCs, the cell quality, and production costs. Due to sky-high costs, the main emergent source of MSCs is allogenic. These are donor-derived MSCs intended for off-the-shelf use for any recipient, instead of using the patient's own stem cells. The reason is intuitive, because of a lower cost of goods. Allogenic cells are cheaper to produce as each batch is enough for multiple more doses for many patients. I will break down the manufacturing process of MSCs into three parts: upstream processing, USP, downstream processing, DSP, and fill and finish. We will only focus on upstream and downstream processing because these are the main parts of MSC manufacturing. Now, the starting stage of the MSC production cycle represents the main cost driver for MSC manufacturing due to the enormous amount of money spent for raw materials. This is called upstream processing. USP. USP is about generation. The goal is to grow as many cells as possible. Scaling up is relatively straightforward. You simply use a bigger bioreactor. The cost and complexity doesn't increase linearly with volume, but costs remain high due to downstream processing bottlenecks. So, in conclusion, there is very good economies of scale to be had here in USP, but the main challenge is in downstream processing. Now, downstream processing, DSP, is about purification. Our goal here is to isolate the fragile MSCs from the massive volume of culture media, microcarriers, and waste that we don't want, but at the same time, keeping MSCs alive and functional. This involves a series of discrete, sequential, and often very slow steps. Many technical bottlenecks exist, which makes it challenging to scale up. For example, centrifugation at present can only be done at a small scale. If we make a centrifuge larger, physics will change and MSCs will be destroyed in the production process. Therefore, it is economically unfeasible to simply expand clean rooms and buy more machines. In addition, DSP steps must be performed within a short time window, limiting the volume we can process at any given time. Let's first begin with upstream processing, USP, where cells are seeded into bioreactors and go through an expansion process. These bioreactors have two important components that we will discuss later: microcarriers and serum. Firstly, single-use bioreactors, or SUBs, are closed stirred tank vessels made from disposable plastic. These are the most popular type of cell expansion method to manufacture MSCs. Cells are inserted and suspended in a culture medium, and for MSCs, small beads called microcarriers are added into the culture medium for the cells to attach onto. For single-use bioreactors, we can split them further into stirred bioreactors, wave-mixed bioreactors, hollow fiber bioreactors, and fixed-bed bioreactors. Among MSC manufacturers, the favorite is the stirred bioreactor. It has the best avoidance of cell damage and cell expansion. It was able to achieve sufficient mass transfer at minimal shear stress. What does shear stress mean? Shear stress simply refers to the friction between layers of fluid in the bioreactor. This acting shear stress is a large part of how we evaluate the performance of the bioreactor because MSCs are very sensitive to shear stress. Planar multi-layer flasks have been traditionally used instead of bioreactors, but they are being phased out in MSC manufacturing because it is harder to scale up, harder to control, and harder to monitor the cell expansion process, despite sometimes being much cheaper for small-scale production. As you can tell, the main difference between a planar flask is that it is two-dimensional. Cells are grown on a flat surface, while an SUB is three-dimensional and cells are suspended in a stirred liquid. Planar multi-layer flasks are like bakery tray racks. They stack multiple cell growth surfaces vertically in a unit, while single-use bioreactors is like a pot of broth and constantly stirred. Stirred bioreactors have pedals, either segment blade impellers or marine impellers, inducing an axial-oriented fluid flow, beneficial for microcarrier-based expansion in the bioreactor. This can be further improved by adapting the impeller diameter, impeller off-bottom clearance, or the impeller blade angle. Today, instrumental stirred single-use bioreactors with sensors are commercially available at small, benchtop, and pilot production scale. They hold a large-scale 150-liter working volume and deliver high cell densities at 2 to the power of 10. They hold a large-scale 150-liter working volume and deliver high cell densities at 2 times 10 to the power of 6 MSCs per ml. Small benchtop scale bioreactors are mainly used to study the MSC production process, and larger ones are used for pilot or production. Microcarriers are beads used in the bioreactor to expand MSCs by providing a free surface area for cells to grow. They are spherical due to their high volume to surface area ratio, and this is their primary advantage over a 2D surface, because when mixed in a bioreactor, they enable a lot more surface area to be used by cells to grow and produce much higher cell densities. Their main role is to ensure that the critical quality attributes CQAs of the MSCs do not change. Manufacturers evaluate microcarriers by their cell attachment efficiency and sensitivity to shear stress. There are many commercially available types of microcarriers from GE Healthcare and Solar Hill for different types of cell expansion needs. Some of them are coated with collagen to improve their ability to bind to cells. Microcarrier surface can also change the phenotype of the produced MSC. Recent studies show that planar structure, material stiffness, nanotopography, and local curvature all impact the proliferation, maintenance of phenotype, and differentiation ability of MSCs. Efforts are now being made to make GMP-grade biodegradable microcarriers and animal-free collagen coatings to improve the MSC batch consistency. Serum must also be added into the culture media. Most culture media contain about 10 to 20% fetal bovine serum. Fetal bovine serum, or FBS, contains proteins and additives like 1. Proteins mediating adhesion to tissue culture treated plastics. 2. Lipids for cellular anabolic purpose. And 3. Growth factors and hormones to stimulate cellular proliferation. There is a disadvantage with FBS because of their potential of contamination. This is because they are sourced from animals instead of humans. They are also expensive. The industry is on the path to transitioning away from FBS towards alternatives like human-derived serum and human platelet lysate, a mixture of growth factors obtained by breaking down human blood platelets. However, the cost to produce these human blood products still remain extremely expensive. Due to high costs, scientists have explored chemically defined media for large-scale manufacturing. Moving on to DSP. downstream processing. It has the largest share of bottlenecks because of the short window of time available to transfer MSCs from culture medium into the cryopreserved state. The difficulty is to do this efficiently and reliably without significant losses in yield, quality, and time. Manufacturers limit production capacity to 150 to 200 liters per batch at present, while upstream processing can easily scale up to 2,000 liters or more than 10 times. Therefore, downstream factors are the limiting factor because of their throughput ability. Cell detachment is then done using proteolytic enzymes like trypsin to break adhesion proteins between cells and microcarriers. The microcarrier surface coating, whether collagen or fibronectin, determines the enzyme's effectiveness. Thus, the main challenge during detachment is ensuring that cells are released without damage. Next, we proceed to cell separation. Cells are separated from microcarrier beads through size exclusion filtration. Manufacturers aim to achieve the higher cell recovery through filters, and the key challenge here will be microcarrier fragmentation, where debris smaller than the filter's pore size are generated during the earlier cell expansion process, leading to contamination of the product as they move through the filters. After this step, we go on to cell washing and cell concentration. At this step, we want to purify the cells by removing impurities. Tangential flow filtration is applied to pump cells across a filter surface, but not directly through the filter surface. Therefore, the cross flow will prevent clogging in the filter membrane and concentrates the cells into a final purified volume. Next, the cell concentrate is processed through continuous flow centrifugation in a single continuous stream of liquid. And while cells are continuously extracted, waste liquid is also extracted. What happens after we have the pure product? We have to characterize the MSC and label what kind of cells we have produced and just how potent are they, so doctors can be assured that they are using cells of standard potency across many patients. But here, there exist numerous difficulties. Firstly, it may be surprising to know that we still don't understand the mechanism of action of MSCs. For example, we still don't really understand how they work therapeutically in the body. And that makes it difficult to know what exactly we need to measure to find out its potency. Second, unlike a simple chemical drug, a cell is more than hundreds of thousands of times larger than a chemical drug. Therefore, the final MSC product is shaped by its production process. Even the same cell, grown in a different bioreactor, can produce different therapeutic effects. In practice, many manufacturers will discuss characterization with regulators first and then choose certain markers to measure through a patent. Because the MSC mechanism of action is not very well known, we are still experimenting with the optimal number of cells per dose for MSC therapies. Therefore, we will adjust MSC dosage based on observed therapeutic effects from early clinical trials, rather than understand the biological properties at work. Naturally, this produces a regulatory hurdle in terms of safety. MSCs are typically made on a 3-month manufacturing timeline, and this long timeline makes it difficult to order a batch of MSCs because patients typically need them urgently. So, after MSCs are manufactured, they are cryopreserved and become an off-the-shelf product with a long shelf life. In conclusion, while upstream processing USP in bioreactors represent the main cost driver due to expensive raw materials, it is downstream processing DSP that acts as the critical bottleneck. DSP struggles with purifying fragile living cells at large scales without damaging them, limiting batch sizes and overall throughput. Compounding these hurdles are difficulties in characterizing the cells and defining doses due to an incomplete understanding of their mechanism of action, creating significant regulatory challenges. I hope that this video have convinced you that the manufacturing challenges of MSC therapies are technical and not so much scientific. Thank you very much. If you are interested, I have included the links for the references in the description below.